Hi, this is Dr. Pedro Ramirez, Editor-in-Chief of the International Journal of Gynecological Cancer. And today I have the great pleasure and honor to have on the podcast Dr. Angeles Alvarez Secord, who is in the Division of Gynecologic Oncology, Department of Obstetrics and Gynecology at Duke School of Medicine, Durham, North Carolina. And the reason for this podcast is the article titled Genomic Alterations, Molecularly Targeted Therapy and Survival, a real-world endometrial cancer molecularly targeted therapy consortium. Angeles, welcome to the podcast. It's so great to see you. It's so great to actually discuss this manuscript with you, and congratulations again to you and all the authors.
Thank you, Pedro. It's so wonderful to be here. And I'm really impressed with how well you said the title. It's quite a mouthful.
Well, quite a compliment. So thank you very, very much. And
Well,
I know that there's many who share that same opinion.
well, it's actually, we've actually just shortened it to ECMT squared because it is a lot of words in there. And you can imagine what it's like talking to a patient about being on this study. They look at you and their eyes keep getting bigger as like you keep talking about the title.
Exactly. So perfect. Thank you for that clarification and for the compliment. So Angeles, obviously, again, this is a very relevant topic, very timely topic. So I wanted to start by discussing as to when and why should tumor testing be done in the setting of endometrial cancer?
Well, fantastic question. I'm sure there's a lot of different opinions. I think testing should be done at the initial diagnosis. And it doesn't have to be expansive testing, but at the very least, mismatch repair and P53 expression. I do recognize some of the costs of these tests, but I think that those two elements provide a tremendous amount of prognostic information and also can tell you how best to treat the patient, both in the advanced and earlier stages of disease.
Excellent. So with that, then can you tell us a little bit about the rationale for this study? What led you and your team to consider that this was a topic that needed exploring?
All right, so dial back a little bit. We go way back, right, Pedro? We did our first interview together in like, what was that, 1997 maybe?
That's right. At
UCLA. So we
UCLA.
we go way, way back. But anyway, my fellowship was at Duke, and under the tremendous leadership of my mentors there, my focus really was ovarian cancer. And then I think around the mid 20 teens in that period of time, just noticed so many more cases of endometrial cancer and younger patients being diagnosed, not just with early stage disease, but advanced disease, and seeing more and more individuals on the in-service as well as dying from these cancers. And it just became something that I felt I needed to start turning my research toward endometrial cancer. Ovarian cancer continues to be a big problem that continues and needs a lot of focus, but it just became so tangible to me, and it really struck home when patients that lived in the neighborhood next to mine were my patients. They became my patients, and I knew them. And one individual, I knew her children, and I just at that moment had this big switch that I needed to do endometrial cancer research. And that's a big deal when your research focus was something different and you don't have any grants in that area to make that change. But I just became incredibly passionate about endometrial cancer and the need to learn more and try to identify ways that we can really help our patients and prevent these cancers from happening. And so that led to this idea, and I guess part of it too was this frustration. We have chemotherapy, we have radiation, and the studies were all about, can we combine them? Do we just do one? And the studies would come back, and for the most part, they were negative, or it may be beneficial to add chemotherapy in this one group. And then GOG 258 was a negative trial where adding radiation to chemotherapy didn't appear to help. And it was almost as if, okay, we're a hammer, chemotherapy and radiation are a hammer, and the whole world's a nail, and that's what we're going to take to everybody, even if it doesn't necessarily work. And how can we tailor therapy to be individualized for the patient and for the abnormal proteins and genes in their cancer? And I was aware of some of the data because we were very lucky at Duke to be part of these studies. And some of these studies were the 86P study. Do you remember the old GOG 86P, which is a 3-arm study of 3 different drugs? And they did translational research, and they found that individuals who had mutations in TSC were more likely to benefit from an mTOR inhibitor. And if they had mutations in beta-catenin, it appeared that it was predictive for efficacy of bevacizumab. And then there was the Amanda Nichols-Fader paper with Dr. Alessandro Santin that evaluated trastuzumab in patients with serous endometrial cancers with HER2 high expression that showed a benefit in PFS. And then there was some of the data about hormonal therapy from Dr. Fiorico with Megace and tamoxifen alternating. And, you know, we tried to do a crossover trial of that compared to chemotherapy and GOG, and it unfortunately couldn't enroll patients. And then we had the approval of immunotherapy in patients who had tumors that had either MSI high or deficient MMR. And those were kind of the new tools on the block. So I thought, what if we could use these targeted therapies and direct treatment based on that? So adding immunotherapy to chemotherapy, adding trastuzumab for patients who had serous cancers with HER2 positivity, adding bevacizumab for patients who had beta-catenin mutations. And now in 2025, it's like, oh, that's not a big deal. Everyone's doing that. But in 2017, that was kind of a new idea. So sorry to go through that long story, but I'm getting there. I promise I'm getting there. So I put together this thing called MODEL, which was molecularly driven endometrial cancer treatment. Don't ask me why model came up. I was really into acronyms at that time. So I talked to my partners about it. Are you laughing at me? I think you're laughing at
No, I love it.
me.
I'm so admiring of the way you're articulating the trajectory of how you started and how you've come to this point. So
Yes.
please.
So I came together with this model idea of how therapy should be based on their individuals' protein expression or gene alterations. And some of my partners were really enthusiastic, others were very cautious. I had some, this really needs to be done in the context of a trial. But like, this is never going to be done on a trial because you can't get all these pharma companies to come on board, and some of these drugs are off patent. It's just not going to happen. So I happened to be sitting at an NRG meeting, and I was sitting next to Dr. Bhavana Pathuri, and we were both sharing our frustration about endometrial cancer. Like, we have to do something. Nothing is really working. And our patients are dying, and we're seeing younger and younger patients being diagnosed. And so I said, well, I have an idea. And she's like, well, I have an idea. And it turned out that our ideas were very similar, but distinct. And so we worked together to put together a trial called EMBATTLE. Again, a wonderful acronym. And it got through one, acceptance at one level at NRG, but then it got declined at the next level. And so then we came up with this idea. It's like, we cannot do this through the clinical trial. We need to put together a group of individuals who are willing to do a real world analysis of this. And there you are. So we came up, and actually, I was very fortunate. I was at Duke and saw this email come through from the Duke Cancer Institute about a Kay Yao Jimmy V Foundation for cancers that affect women. So the idea was to put together this real world database of a multi-center institutions. So there's 9 founding institutions that started this off. And thank you to my friends who were willing to do this with me. For the most part, everybody said yes, but we did have some no's. And it takes a lot to say yes, right? Because even though we got some money, it wasn't a tremendous amount of money, and it was a lot of work. And it's the first time of doing something, so you just don't know how well this is going to turn out. But we had 9 founding sites, and our big idea here was, number one, we had to show that there was genomic alterations in endometrial cancer. Because in 2017, not a lot of people believed that there was actionable mutations in endometrial cancer. So that was our goal. We had some data that we did with the foundation team. Dr. Julia Elvin worked with us, and we found that over 80% of those specimens had a targeted alteration. And so we had some preliminary data. So number one actually was just making this consortium happen, bringing together 9 different groups of individuals. You can imagine all the contracts for the data transfer agreements, the material transfer agreements. And then the other part was, what is the frequency of genomic alterations? And then the last part is match therapies going to have a higher response rate, improved survival outcomes compared to traditional chemotherapy. So that's how it all happened, Pedro.
What an amazing, inspiring trajectory. And I'm so glad you share that, because obviously we have a lot of young listeners who are obviously either trainees or young faculty in gynecologic oncology. So Really, really very, very impressive. Congratulations. So let's talk a little bit more about that. So how what would you say was the methodology for this approach? And you know, and ideally again, obviously share the results and the findings that you want to highlight to our audience.
Well, methodologies, challenging with a real world study. Real world studies are going to be dirty. They're not going to be perfect because you don't have a lot of robust control over the data. And so a lot of the decisions that were made by the treating physicians. It's not like you have a cookbook clinical trial. So the tumor testing, type of testing done, whether it was IHC, PCR, or next generation sequencing, and whoever was utilized for the sequencing, that was all physician based. The type of treatment was physician based. So it wasn't as if the type of match therapy was something that we constructed that people had to follow. We did have the model algorithm in there so they could say somebody was treated per model and it would say, HER2 positive, so they received trastuzumab. So that's just an example. Deficient MMR received immunotherapy, but there was no control over that. Those were all decisions that were made based on appropriate clinical care. The really hard part here was just even setting up the data abstraction. So you have 9 different groups. All of us have different Excel files. We did ask everyone to share their Excel databases with us, and that was really helpful. And then we had to merge them. And then we sent it out for everybody to review and to provide any changes. And then we put together the database and had run through it with some test cases and thought it was okay to go. And the devil's in the details. Every step of the way, you uncover something that it seems so clear to me, but somebody else looking at it was like, "What on earth does this mean?" All right, so we get our grant in 2019. What happens in 2020? COVID hit. So one of the things that we wanted to do to make sure the data was clean is have our coordinator at Duke go and travel to these sites and actually review all the data personally to make sure the data entry was correct. So COVID hits, can't do that. Everything gets shut down. So we figured out, well, they could send us the data, the redacted files, but it turned out that was too much work for everyone to do. And so we leaned on, okay, well, at least send us the pathology reports and the tumor testing so we can confirm that. We're going to query all the data if it doesn't make sense, like if dates don't make sense. We're going to send you a query and you can review it. And then for the match cases, since we can't go on site and look at your data, please have a second set of eyes review all the match cases to make sure that this is correct. So we did the best that we could to make the data as clean as possible. But, it's a real world study. It's going to have those limitations. All right. So what do we go to next? Well, so we wanted to have a certain number of cases in the database, and we had over 1,000. And when we looked at those 1,000, and we felt that not all of them, either we couldn't confirm their records or queries weren't addressed, we dropped those cases. So we had the 967 left is where we felt confident about the data. One of the really exciting things for us, and at that point when we published this, we had expanded from 9 to 12 centers, is the patient composition. One of our concerns with clinical trials is that often Black patients are not represented. And you see this in a lot of different studies where at the time when this study came out, maybe about 4 to 5% of the patients who were enrolled on trials were Black. And yet that's the area of really tremendous need. There's need across the board for everyone, right? But when you have patients who identify as Black being at twofold higher risk for death, I think the urgency, that sense of urgency is heightened. And I was really excited that in our study, 24% of the patients identified as Black. I think is really important because as you try to unpeel back what's going on with endometrial cancer, it goes back to what is the underlying biology of the disease. And if you don't have enough patients that represent the disease biology, you may be identifying treatments that work great in one group, but really aren't going to work great in a diverse patient population. And we also wanted to assess better what is driving these cancers. And in our patients, we also identified that over 90% had at least one alteration. And we when you do a grant, you can't go on a fishing expedition. It's like if you want to not get a grant, you have a fishing expedition as I'm going to study NGS across all these endometrial cancers. You have to be very concise. And so our initial grant actually didn't even have 11. We expanded our statistical analysis of plan with time, but I think our initial grant was really, I think it was P53 and it might have been HER2 and beta-catenin, but it was very small. And then we later expanded to include like ERBB2 and ERBB3. And I can't remember if we had the mTOR P10 AKT pathway in there initially. But, just really, that's a very important thing when you're writing a grant. You want to limit it to a focused area of interest. You can always do the study and then do additional data mining, but don't put like the fishing expedition as your primary objective. And then I was really thrilled to have at least 233 patients that received matched biologic therapies. And at that time, remember, matched biologic therapies either weren't available or they weren't FDA approved for that use, or they hadn't yet had NCCN a listing. And so the number and matched therapies may not match because these patients didn't just start being put into the database in 2020 or 2021. They may have been patients that were diagnosed earlier too. And in that group of patients that had matched biologic therapies, we identified that the progression-free survival was 6.9 months and the overall survival was 20.5 months, which was longer than what you'd expect with traditional chemotherapy. And I hope I synthesized that in terms of the nuts and bolts of
Yeah. No,
methodology.
this is fantastic. And I'm really glad actually that you brought up the issue with regards to a patient population that is often not represented in trials. And I'm really glad that as we move forward, there is a focus on assuring that we as investigators really try to recruit patients that traditionally have not been included. So thank you for outlining that. So Angeles, we routinely in the podcast, we have questions from the fellows in our journal. This first question comes from Clara Santia, who's with us at Houston Methodist. She's from Buenos Aires, Argentina. And she's asking, could you share with us how you picked specifically the 11 pre-specified genes of interest?
Yes, absolutely. That's a fantastic question. So you know, we want to look at all these genes, right? And actually, that's a focus of one of our other papers. But this initial rollout, we wanted to focus on genes that we felt could be actionable for a treatment. So PI3K, P10, those are genes, and AKT, those are genes that may portend a benefit to a treatment with that mTOR inhibitor. P53, in part because of associations between P53 and higher efficacy with bevacizumab, and that came out of the GOG 86P translational data. ESR1 is a very interesting gene. My partner at that time, Dr. Stephanie Gaillard, she was involved in this gene, specifically in breast cancer research, but this is a gene that if it's abnormal, there's an alteration, it can alter how patients respond to hormonal therapy. And then mTOR and TST2 are also involved with drugs that target that pathway, so mTOR inhibitors. The pole mutation, that was something that came later that we added on because of the molecular classification and identification that that gene was very important in outcomes and what is the role of that gene in patients who have immunotherapy. And then ERBB2 and 3 because of the link with HER2 expression and potentially higher responses for patients who have treatment with HER2 with targeted agents against HER2.
Great. This next question is from Julia Gellison. She's at the University of Wisconsin in Madison. And she's asking, were you surprised to find such high rates of genomic alterations in the 11 pre-specified genes in this population?
Really, I wasn't because in our initial preliminary data with Foundation, I think it was over 80%, and that was even a more limited set of genes. So it really didn't shock me too much. I was happy to see that.
Yeah. This next question comes to us from Matteo Marchetti in the University of Padua in Italy. He's asking, one striking finding from the study is that although over 90% of patients had at least one potential targetable alteration, only about a quarter actually received the match therapy. In your view, what are the main barriers to translating molecular data into real-world treatment decisions? Are we talking about cost, turnaround times for sequencing, or limited access to drugs and regulatory constraints?
Great question and great answer. All of the above. Um, this is again a real world study where the treating physician made decisions regarding the treatment that patients received. And during the time interval as well, some of these treatments may not all have been eligible. One of the things that happened during the course of the study is pembrolizumab had a treatment as a tumor agnostic indication, but Pamlin as a combination wasn't available until years after that. So while some of these patients did receive Pamlin, maybe that wasn't available to all of them.
Yeah. Christian Brown from Lucerne Hospital, he's asking, were there any instances where combination targeted therapy were used in patients with multiple actionable genomic alterations? And if not, was there any analysis on how physicians decided which alteration to prioritize when selecting the therapy?
So this is, and I love this question because I'm like, ooh, I want to go back and look at that now. Um, I'm absolutely sure that there was a case where people had multiple genomic alterations. The key here, though, is are they all actionable? Because not just because you have a genomic alteration doesn't necessarily mean it's actionable or going to make a difference. And that's where we need to do more research. Again, there was no control over the selection of the treatment or sequencing. But I do think it's worthwhile to dive back into that group and see how many of those individuals could have been a candidate for more than one therapy. But I know from treating these patients in clinic, often there's more than one therapy that I can choose for them, especially for individuals who have endometrioid type cancers.
Yeah, excellent. Now let's get into some questions about oncologic outcomes. And Stephanie Gill from University of Toronto, she's asking, can you elaborate on why you think the progression-free survival at 6 months and median progression-free survival were similar for immunotherapy, non-immunotherapy regimens, but then became significantly different at 1 and 3 years? Do you think there's a crossover of treatment that was received and not captured?
So I'm going to dive back on this question a little bit because I think it's important to note again when this study was developed to where we are in this current era. And what's so exciting, Pedro, when you and I first started, you could read a textbook and you didn't really have to read anything else for several years because things didn't really change too much. You could keep the same slide carousel. Oh my gosh, we did use slide carousels, it's true. For way over a year and maybe have to change a slide or two. But now the speed of which things are changing so quickly is I'm completely revamping a slide deck from meeting to meeting. We have so much data and groundbreaking research coming through. So my point that I wanted to make is when we added this part on about evaluating immunotherapy versus the non-immunotherapy groups, because we anticipated that reviewers would think, well, your findings are really due to immunotherapy and you can't really state whether there's any benefit of other match types of therapies. So we amended our statistical analysis plan to add in this subset analysis, specifically for the match therapies grouped as non-immunotherapy and immunotherapy. And I actually thought going into this analysis that the PFS rate at 6 months would be the same for both. I was actually very pleasantly surprised to see that it was similar. It was approximately 54% for each of these groups. However, what we do see happening is that the progression-free survival rates are much higher for immunotherapy compared to the non-immunotherapy. But what does that mean right now? We have to dive into this more. Does it mean that immunotherapy should be given first before any other biologic match therapy? That's certainly a hypothesis, but I don't think we can prove it with this study. I think that's something that needs to be very carefully assessed in the future because we have more treatments now. We have the ADCs coming up, which are super exciting. So if somebody is eligible for an immunotherapy versus, for instance, trastuzumab, which one do you use first in the advanced or recurrent setting? I don't have a great answer for that. In fact, I don't even want to answer that because I know that it'll be unpopular, whatever I say on both sides of this argument. It's a great debate.
That's right. Exactly. Well, no, excellent point. And now let's talk a little bit about this unique population with very good outcomes. Thomas Bartel, Medical University of Vienna, he's asking, the authors in this study report on 10 recurrent endometrial cancer cases harboring a poly mutation, which is an impressive number. Do you have more detailed information available regarding the recurrence therapies and respective responses in these poly mutant cases? I like to emphasize that this subset may hold significant value and potentially warrant a separate publication. So therefore, a recommendation from one of our fellows.
Yes, thank you so much for that recommendation. It is underway. Honestly, I was surprised by the low rate of pole alterations in our group. And because it was really only about 2%. I think part of this goes back to our patient population. Those initial studies with pole mutations may reflect more of the patient population with a higher frequency of individuals who identify as white compared to in our patient mix, 24% identified as Black. And in terms of the outcomes, not everybody that had a pole mutation in our study had a good outcome. So absolutely, you are correct in that this needs to be another paper that's written.
Excellent. So you alluded a little bit to this question from Christian Brown. He says, as new targeted therapies became available over the course of your study, was there any assessment as to how these changes shaped the clinical decision making in the routine practice?
Oh, absolutely shaped it. I mean, the biggest one was the use of pembrolizumab and lenvatinib for patients who don't have deficient MMR or high MSI. This is incredibly important because I think this is a situation where you have a negative biomarker is how you can direct therapy. So even though proficient mismatch repair wouldn't necessarily be seen as abnormal finding, it is still an important finding that can be used to determine your management decision. The other areas of interest too is we did have some patients who were treated on clinical trials. So there was, I think, a patient who received a ROS inhibitor. There was also some use of trametinib in patients who had HRAS or KRAS mutations. I think RAS mutations are of definite interest in a lot of different cancers, and there's newer drugs targeting RAS mutation. I'm very interested in those. And we had a patient who was treated with a WEE1 inhibitor who had a TP53 mutation. And that was over the course of that short period of time. We now have over 3,800 patients in the database, so it would be great to take a look at this again.
Fantastic. So this next question goes back to your point about ethnicity. Thomas Bartle is asking, did your analysis reveal any differences in mutational landscape and or molecular tumor subtypes between patients of different self-reported ethnic backgrounds?
All right, fabulous. This is a great question because this has been done now. It was reported by it's a sub-study of our consortium, and the work was done at UNC by Dr. Vicky Bay Jump and Dr. Glenn Boyles, and it was presented at the Winter SGO meeting. And they were able to find that there are two distinct mutations that seem to be more frequent in patients who identify as Black compared to white, but it is incredibly rare. I think it was BCL2 and it may have been FGF, I think R3, I think. I have to double check. So it was just recently reported, and we're looking forward to the publication.
Very interesting. Um, Stephanie Gill from University of Toronto, she's asking, you included carcinosarcomas. Did you specifically look at the tumor profiling for those with carcinosarcoma and see did this also correlate with race?
We have not done that sub-study yet. However, Stephanie, feel free to reach out to me because anybody who wants to be part of this consortium, we welcome you all.
She'll definitely will, and she'll be glad to hear that as well. So, Manuel Montesinos Albert from Valencia in Spain, he's asking, given the variability of testing platforms and the decentralized nature of this study, how do we begin to create more uniform standard around genomic reporting and therapeutic alignment in endometrial cancer?
That would be really tough because there's all of these different companies, right, in this space. The NGS testing, probably not too problematic there. I don't think there's a lot of differences in terms of how they report the actual gene alterations. However, one area that's really tough is TMB. There can be a lot of differences in tumor mutational burden because of the different testing methods, and a single tumor mutational burden threshold really can't be applied to all the assays. So that's one where you have to be much more careful.
So now, I want to ask you the limitations of the study. What do you see as the limitations in a study like this?
Anytime you do a real world study, there's going to be limitations. As we talked about before, I can always say that, this data is dirty. Even if you try to clean it, it's not going to be robust like a clinical trial or where you have monitors coming in. So you just have to accept it. And the limitations here are going to include bias. So is there a bias toward including just patients who had testing on this study, or a bias regarding the inclusion of patients who had matched therapy but did well, right? The use of self-reported race is, and we don't even know if it's self-reported sometimes. We've gotten it from the electronic medical record. So this assumption, this is how the patient self-reported their race. There's several of the questions here were brought up about the treatment decision. So there's non-uniform treatment and follow-up. There's a lack of centralized pathology and radiology review, sampling bias, heterogeneity in treatment groups, lack of adjustments for multiple comparisons, you name it. And this is mostly all academic centers. So you have to limit some of the generalizability of the study to academic standards. I don't know what this would look like in community practice, but I do think, working, because I work at a community practice as well, as part of my work at Duke. And the medical oncologists are very savvy about utilizing matched individualized therapy. They've been doing it for a long time, much longer than us.
Yeah, yeah, absolutely. I experienced the same in my own institution as well. So Angeles, I just have two additional questions. This one's from Matteo Marchetti, and he's asking, do you think this integration is relevant only in the setting of advanced or recurrent metastatic disease, or could it eventually also apply earlier in the course of the disease?
I think it could be applied earlier. I think that's an area where we still need a lot of intensive research. But just imagine, a point in time where you've identified the driver of that cancer and you can target it with that drug and you can make it all just melt away. So, not, even if somebody has early stage disease, we're treating them with adjuvant therapy, but we don't even 100% know if it's truly going to help them,
Hmm,
right?
absolutely,
So
absolutely.
if we can target that therapy better, especially those patients who have high risk disease like the serous cancers, can we target that in a way that's feasible and mitigate toxicity as much as possible? Because those patients don't have any symptoms from their cancer. So we want to be really careful that our treatments are not overly toxic.
So Angelos, as a last question, and you mentioned the fact that obviously there's a field that is evolving so quickly. Um, Given your expertise, your experience, where do you see this going? What are the potentials for integration of molecular classification, molecular analyses in our therapy?
Oh, I want to cure our patients. I do. I want to cure all of them. And I want to prevent this cancer from even happening. We've just seen this explosion of endometrial cancer, and it breaks my heart, especially in younger patients. And there's, right now, Pedro, we're doing a lot of research on patients who have EIN, or the precursor to endometrial cancer, as well as early stage endometrial cancer. How can molecular classification help us there? But it's even beyond those four molecular classifiers. I think this is a much more complex disease, and the simplified classification's not really going to comprehensively help us understand it, but we've made tremendous inroads. So in terms of your bigger question, where do I want to go from this? I want to cure all our patients.
What a wonderful way to end this podcast. Thank you so much, Angeles. I know you're incredibly busy. Thank you for accepting our invitation. I'm so looking forward to the journal club on this topic. And I really do want to thank you again for all of the contributions you have made through the course of your career as a gynecologic oncologist. Continue to make through that course. And I think very importantly, for the role model that you are to many.
Oh, that was so kind of you. Thanks, Pedro. I mean, I feel I can say all that about you as well. I mean, look at all the great accomplishments you've made over the time of your career. This was just an absolute pleasure to be with you this afternoon and be able to share this story.